Hi everyone, it's Jeff Gruen. Before I get started, Yale University requires I disclose my conflict of interest, which regards a provisional patent describing the association between the gene I'm going to talk about today and decoding performance. I'm an inventor on this application as well as other members of my lab. So with that, I'd like to present to you early results from our New Haven Lexinome project and give you our fall 2020 update. I'm going to start with a short video from one of our intervention programs at a local elementary school within the New Haven Lexinome project.
First, we're going to peel off non at the beginning of the word. Now look at this. It's shoal. Shoal. What does it say? Shoal. So I'm going to peel off shoal. Shoal. My root is so. My whole word is not social. It's whole word shoal. Not social.
So these four children are really struggling with reading. As you can see, worldwide, about 10% of students actually struggle with reading. These are students in regular classes, at regular schools, and with normal and above normal IQs. Between 60 to 80% of reading performance is due to multiple genetic variants, making reading a highly heritable trait. The goal of the New Haven Lexinome project is to find out the extent to which genetic variants correspond with response to intervention through whole genome sequence analysis, behavioral testing, and functional MRI studies. We've recruited and studied about 500 children, and each of these children have undergone serial cognitive batteries that assess reading, language, executive function from grade 1 all the way through grade 5. In addition, each one of our kids have given us saliva samples for which we've done 30x whole genome sequencing. I'd like to present to you our genome-wide association study, our initial study predicated on a decoding composite score, which really consists of 5 mainstream reading assessment subtests: a word attack, which is decoding, word efficiency, and as well as comprehension. Our GWAS has been adjusted for age, race, ethnicity, sex, and socioeconomic status. And here is a typical, and this is our results presented as a typical GWAS, a Manhattan plot. The chromosome left markers presented here on the left, chromosome 21, our smallest chromosome here on the right, or chromosome 22 as well. And our thresholds for p-values that are graphed here on the y-axis. And what you can see here is the red axis is the genome-wide threshold, or Bonferroni threshold at 10 to the -8th. And you can see a single peak of markers from a single chromosome, chromosome 19, that correspond with an association with our decoding performance. So the first question we need to ask is could these results be due to error from unaccounted population stratification? And to do that, we look first at our QQ plot. We look at all the markers, what we expect to see them, what we actually observe them to see them. For the pretty much it observes a straight line. So it doesn't appear to be any genomic inflation factor. In fact, it's less than 0.01, 0.06, which is fine. And there's a single peak here, which happens to correspond nicely with our single peak on chromosome 19. We also looked at the self-reported ancestry from each of our families. And for the most part, they report African descent, European descent, and Hispanic ancestries. And we map them on to the superpopulations from the 1000 Genome Project. Here's the European group, the African superpopulation, and the Hispanic superpopulation. And then we mapped the genotypes from our New Haven Lexinome Project children onto these. And you can see they pretty much follow this pattern. And so we're able to control for population stratification with pretty much with 10 principal components. Actually with 2, we expanded it out to 10 anyways. So we reviewed our QQ plot. We mapped our New Haven Lexinome projects on 1000 genome superpopulation control with PCs. We also added controls for socioeconomic status in our populations and in data. I didn't show you. We showed that allele frequencies for our hits are the same in our subpopulation as well as our decoding performance. We also replicated our results in the GRAD study, which is about 1,350 African Americans and Hispanic Americans, all with similar phenotyping. And so that brings us to the next. So I think that's pretty reasonable that we've accounted for all the population stratification. The next question is, what gene corresponds with this peak of association here in chromosome 19? And to do that, we can look here in depth at our genome browser, and we can see here is in higher magnification is all our markers fall into a small genomic region of about 200,000 bases on chromosome 19, and identify 4 genes that are encoded there. But most of the markers here shown in this level fall on this gene called KIAA 0355, and here it actually is the peak. Regardless, we did check the expression level, and when we look at expression in GTEx, it shows that KIAA 0355, or a gene with the largest number of markers on it, actually has very significant brain expression, but interestingly, very high expression in the cerebellum. And it's interesting because it supports the cerebellar theory of reading disability, which was something proposed in the 1990s by Nicholson and Fawcett, because reading disability has been associated with deficits in cerebellar-related functions such as dystonia, time estimation, skill automatization, and balance. And what they proposed was a cortical cerebellar circuit that has an important role in reading and reading development. So I started out this talk with presenting 4 of our kids in one of our intervention program and showing you that real difficult time reading. And then I showed you our GWAS results from our entire cohort. And now it's time to bring back our data to the longitudinal data that's generated and bring it back to the classroom. And when we look at our individual assessments of reading, when we look at decoding as letter word ID, we look at comprehension, bottom left-hand corner, again, decoding, and reading fluency, what we can see here, represented in orange, are the children with the KIAA 0355 risk allele. Not only are they performing significantly less at each grade, beginning grade 1 all the way through grade 5, throughout their elementary school career, at each level, at each of these 4 reading performance scores, it's consistent all the way through. They never catch up to their control counterparts. So this has a pretty profound association all the way through in our longitudinal data. And it's an agreement at that age group with the language reading continuum. Those of you who have children know that oral language develops in years 1 through 3. And in fact, that is the equivalent of reading in the pre-reading years. And then the next emphasis really is on print knowledge, in which we have letter word ID. That is, you know, the name of the letters and the sounds that they provide. And then in our age group, we're learning to read. And here the emphasis on phonological awareness and phonics and fluency. And that pretty much coincides with the expression and the association data that we're seeing for KIAA 0355. So with that, I will end my talk. These are the wonderful people that help make it happen. These are the nice folks who provide the support to the ability to do this. And I look forward to hearing questions from you.